In order to figure out how reproduction works at the cellular level, scientists studied frogs in pants and then sea urchins. Old Antony van Leeuwenhoek first observed sperm under a microscope in 1677, but nobody really knew much about reproduction at all except for that there was sperm and that there was eggs. But a popular idea at the time was called Preformationism, which was the idea that there are teeny tiny little humans inside sperm or eggs, and that was the same for other animals too. Preformationists came in two camps, spermists and ovists. Spermists thought that the little tiny person was inside of a sperm, and ovists thought that the little tiny person was inside of an ova. So Lazaro Spazi here was a diehard ovist, Catholic priest, biology type guy. And in the 1760s, he decided to reproduce a reproduction experiment using frogs in contraceptive pants. He collected a bunch of frogs, separated them by sex, and put some of the males in pants. Then the tight pant-clad frogs went out with the females to try to make some frog babies. When the males tried to fertilize the eggs, their spunk got caught in their trousers and no tadpoles emerged from the eggs. But when he collected the frog sperm from the pants and bathed eggs in them, tadpoles did develop. It was the first example of in vitro fertilization, and it provided evidence that both sperm and egg are necessary for reproduction. But despite his contradictory results, Spallanzani remained an ovist. He thought it was the liquid semen portion of the emission that activated the growth of the egg and not the gelatinous spermy portion. So in 1876, Oscar Hertwig here was finally the first person to witness the actual moment of sexual reproduction in an organism. He was working with sea urchins, which are actually a really great model organism for developmental biology. Their eggs are super transparent, and a single sea urchin can pump out shitloads of eggs. He peeped the first moment an urchin sperm fused with an urchin egg, and he saw that it was only one sperm that made it, but many tried. In fact, after one sperm squiggled into there, it formed what's called a vitelline membrane, which prevented other sperm from sperming on into there.